Hello everyone, my name is Ricardo Espinoza Lima. I know it's kind of long. My major is biology, biochemistry. My mentor is Marcus Merck, and I'm working with Max Wilson in the Department of Molecular, Cellular, and Developmental Biology. My project this summer is the signal dynamics of SMAD proteins. But who cares about SMAD proteins? Why should we care about them? In other words, why are SMADs important? I like to divide their importance in 3 parts. First, they help the cell to regulate their growth. So SMAD proteins are actually gonna stop the growing of cells when they're mature enough. Also, if there's something wrong with the cell, the DNA, or there's some part of the cell that is wrong, the SMAD proteins are gonna signal the cell to die or to commit suicide. And the focus of the lab is differentiation. And differentiation is this unique ability of stem cells to transform into any type of cell in the human body. So stem cells are basically a blank cell. And SMAD proteins are going to help the cell decide whether or not to be a neuron cell, a heart cell, or an intestine cell, just to name a few. My focus this summer is to figure out what are the dynamics of SMAD proteins. And first, I'd like to introduce you to this family of proteins. And there are in total 8 SMAD proteins, but 5 of them are going to be the most active, and they're going to interact with the cell more intimately. So they're actually going to reach the cell's nucleus, and they're going to help make genes. They're going to help the cells to make these decisions that I mentioned earlier, to die, to stop growing, or to differentiate if it's a stem cell. But how do these proteins interact with each other? Just like any family, we have favorite siblings. And with SMAD proteins, the family of SMAD proteins is not the exception. So we found out that SMAD 2 and 3, they like to be together, as well as SMAD 1, 5, and 8. But what happens if they get separated? We want to analyze these dynamics. My plan to study the SMAD proteins is divided in 3 steps. The first one is that we're going to clone the SMAD proteins. So the first step is to look at SMAD2, and we're going to make a lot of them. Then we're going to create a backbone or a skeleton for the SMAD protein with a fluorescent protein. So here we have the SMAD2 without the fluorescent protein, so we cannot see them yet. And once we put them and we make the backbone or the skeleton, it's going to light up. The third step is to put the SMAD proteins into the cell and watch their dynamics. So here we have the fluorescently tagged SMAD2 protein, and we're going to put them into kidney cells. We're not going to put them in stem cells yet because the SMAD proteins, their dynamics are conserved. Meaning that how they move and how they interact with the cell should be similar or identical in other cells. Just as we speak, you have SMAD cells all around your body making these decisions. And of course, we need to repeat these steps for the other SMADs. My progress in the project is that we're still in the second step. And this is because we've been having difficulties cloning the backbone, which is the skeleton of the SMAD. This backbone has the fluorescent protein, and the fluorescent protein is right here. As you can see, the circle is not complete, because ultimately we want to just stick the SMAD2 right there to complete the circle. I'm proud to announce that, I've been talking about that we've been having difficulties with cloning the backbone, but how do we determine if it worked or if it didn't work? Well, we use this technique called electrophoresis. And the way I like to think about electrophoresis is an obstacle course. Here we have 2 participants, the red one, which is tall, and the short one, which is blue. Once we put him to just run the obstacle course, we expect the shorter one to travel the furthest through the obstacle course, whereas the tall one is going to have difficulties running through the obstacle course. We can use the same analogy or the same idea with electrophoresis. Instead of an obstacle course, we have a gel. And here we have the same 2 participants, the backbone, which is big, and the SMAD, which is small. Over a certain period of time, we should expect the backbone to move a little bit and the SMAD protein to travel before this. In the middle one, we have markings that tell us the size, and that's how we are able to identify if we actually clone them successfully. And I'm proud to announce that we successfully cloned a SMAD protein. And on your right, you have the SMAD2, and on the left, you have a backbone. So here's the SMAD2. And this is the 1,000 line mark, and this confirms that we cloned the SMAD2. What about the backbone? We expected a horizontal line around here, but we didn't get anything. So this lack of the 10,000 line mark says that there is no backbone. What about the other SMADs? So far, we've been able to also clone SMAD1. And in this picture shows it. I know it's really, really hard to see, but here's the SMAD1. And this is the 1,500 line mark, and this confirms that we cloned SMAD1. We had some interesting results of the backbone though. Here in this picture, we can see that we cloned something in the backbone. But what did we clone? We cloned something, but we don't know what. Well, we just cloned some part of the whole circle, but we need the whole circle, or like the most we can, because everything like in the circle we have important stuff for the project. After many tries, we were able to clone successfully the backbone. But we're still cloning something else. Although it would be interesting to see what we actually clone, or we clone as well. That's out of the focus of this project and requires further testing. Now that we have the ingredients to make the full circle, this is roughly how it should look like. We have this SMAD2 right here, and with the help of the backbone and the fluorescent protein, we are going to be able to make the full circle. But we haven't physically separate. How do we make the full circle? Do we just put them in a flask and then shake it and hope they make circles? Well, it's more complex than that. When we clone them, we make complementary sites to assemble them. As you can see here, we have these spaghetti-like DNAs, and they have complementary sites, so they have common colors in this case. So just as any game, we're just gonna connect all the colors so that we can make the full circle. And this should give us the whole circle DNA that we're looking for. But what about next? Now that we've successfully cloned the SMAD protein with the backbone, we're gonna put them into cells. And what are we expecting to get from cells in these SMAD proteins? Well, this is a simulation of what should we expect. We have a cell in the time 0, and after some time we should start seeing the cell to light up around the nucleus. And then after a while, these little dots are going to start moving into the nucleus. But we already know that. So how does my project fit into this larger scale? The scientific community already knows this. But we're going to be able to answer one of the most important questions, which is, how long does this process take? And by knowing that, we're going to be able to start messing with the SMAD behaviors. And that's one of the future projects. We hope that we can make use of optogenetics to control the behaviors of MADs. And this is a rough diagram of how they should look like. We have 2 proteins of interest, and by using light, we can make different combinations of SMAD proteins, and as well, we can separate them. As an ending note, I'd like to thank my principal and base here, Max Wilson, my lab mentor, Marcus Murr, and of course, CSEP, the EUREKA program, and the coordinators. Thank you very much.